So we continue the conversation about bacterial structures, and the next one, if you would look at the order as I go from the inside, nucleoid, cytoplasm, stuff like that, to outside, cell membrane, cell wall, and now we get to the structure that is called GLYCOCALYX. Sorry about that. Okay, GLYCOCALYX is the layer of carbohydrates. This image, it is shown in green. GLYCOCALYX isn't found in all bacterial cells, but it is found in many. There are several types of that GLYCOCALYX layer that can exist in bacteria. First type, perhaps the most important from clinical standpoint, is called a capsule. Here on this electron micrograph, light micrograph, you can see the purple bacterial cells surrounded by the white layer, capsule. Capsule is sort of a rigid layer that protects the cell from phagocytosis. First and foremost, phagocytosis is a process in which your immune cells, human immune cells, consume and destroy bacteria. So capsule prevents this from happening, making these cells more resistant, these bacterial cells more resistant to your immune system. Does that make sense to you? Now, another important feature of capsule is its ability to increase adhesion to the surfaces. It means that bacteria, once it's inside the body, has better chances to stick, say, to the surface of a gut, surface of a lung, or surface of the cardiac endothelium. You can imagine that both increased adhesion and protection against phagocytosis, they will increase the ability of bacteria to cause disease. I want to let it sink in. Do you understand what I'm saying? So, capsule will make bacteria more virulent, more potent in terms of the disease. Is that understood? Now, I'm going to introduce this term. We're going to talk about these terms later, but I'm going to use it over and over because it's such a common term. Anything that increases virulence, increases a disease potency of a microorganism is called a virulence factor. Now, look at this. Capsule increases adhesion. Is that a virulence factor? Yes, it is. Resistance to phagocytosis. Is that a virulence factor? Yes, it is. Now, not a tricky question, but having a round shape, is that a virulence factor? No, not really. It's just, having a round shape. It doesn't contribute to disease in any way. Now, being resistant to antibiotics. Yes. Does that make sense? Anything that increases the disease potency is a virulence factor. And here's the link to what we talked about. I told you about the protein expression, right? Capsule, a layer of carbohydrates. What produces carbohydrates? Enzymes. Enzymes are proteins. So basically, the ability to have a capsule, the capsule itself, is a trait that is somehow encoded in the genome, in the genes, right? So some bacteria have these genes that encode a virulence factor such as capsule, such as adhesion molecules, such as resistance to antibiotics. Does that make sense? Virulence factors are genetic. Okay? Another type of GLYCOCALYX is slime layer. It's looser than a capsule. Okay? It doesn't protect against phagocytosis that much, but it does increase adhesion as well, and that increased adhesion to the surfaces, either by capsule or slime layer, contributes to the formation of so-called bacterial biofilms. If you don't brush your teeth for a while, and then you touch them with your tongue, how does it feel? Like they have like a little film on them, a little layer of something, right? Your teeth have a layer of bacteria on them all the time. It's a normal bacterial biofilm. Bacteria in the biofilm exist in mutually beneficial relationships. They can be mutualistic, they can be synergistic. They can be mandatory and non-mandatory. But the important thing is that these bacteria enhance each other's growth. So far, that makes sense? And they basically work together, bacteria and a biofilm. And biofilms, the formation of a biofilm prevents the bacteria from drying out, prevents them from being phagocytosed. It even reduces bacterial sensitivity to drugs. So biofilms are notoriously hard to remove. When you brush your teeth, what you essentially do is you disturbing the bacterial biofilm. You don't remove them, no, but you wreak havoc on that biofilm. Say you brush your teeth at 7 a.m. By 7, 8, 9 p.m., the bacteria on your teeth kind of restored it, and there you go, you brush again. So you constantly prevent biofilm from growing further. Does that make sense? If you will not, they will form perfectly resistant biofilm. It will be really hard to get rid of it. They will start to produce different acid that will cause them caries, that will cause inflammation in the periodontal sulcus, and so on and so forth. Okay? By brushing your teeth, you disturb bacteria. Another important case for clinically relevant biofilms are bacterial biofilms on the prosthetic joints. It is incredibly hard to keep absolute 100% sterility, so often joint replacements are contaminated with microorganisms. And if these microorganisms on the joint form a biofilm, that is it. Often it leads to massive infection of the joint, and the prosthetic needs to be replaced. So patients who undergo that surgery often are treated with antibiotics for an extended period of time just to keep microorganisms from forming biofilm. And that's a separate branch sort of clinical microbiology, how to remove biofilms from prosthetic joints that are already implanted. And there was a manuscript showing that you can do it using electricity. So scientists, they got a joint, created a biofilm, and then ran weak electric current through that metal prosthesis, and it destroyed the biofilm. Of course, they do not suggest to electrocute patients to get rid of the biofilm. The idea is how to deliver electricity there. If you can, you're all set. That makes sense? So biofilms are really clinically important. Some bacteria and archaea form S layers. We have really no idea what they do. Probably some structural functions, maybe increasing the rigidity of the envelope. movement attachment, things. Flagella, fimbriae, pili. Flagellum, an external appendage. Yes, okay, good. Flagellum, an external, I just thought something is weird with this. No. It's a long filament that is inserted into a hook, and hook is set on the rod. So how does flagella work? Rod right here or here, it rotates. As it rotates the hook. When hook rotates, flagellum rotates. So it's like a propeller in the back of bacteria. Does that make sense? Now, what I want you to know about the assembly of flagellum, the only thing is that it's rod, hook, and filament. Period. You can see that an actual motor, and this is called a motor, is very, very complicated. Like in extremely, there are a lot of different proteins that contribute and participate in rotating the rod. I want you to know only rod, hook, and filament, and that it works like a propeller. Why it's important? Because bacterial flagellum is a propeller running ahead of a train. Eukaryotic is an oar. You see the difference? Eukaryotic flagellum waves, bacterial flagellum rotates. Flagella can be arranged in a different way. This is something else that I will ask you about. They can be monotrichous, one flagellum at the tip of the cell. Amphitrichous, flagella on both cellular poles. Lophotrichous, a bunch of flagella at one end. And peritrichous, flagella all over the cell. Any questions about this? You have to remember those. I give you a description, you give me a name. Okay? Fimbriae. Here's the brush of short proteins, okay, on the surface of the cell. Fimbriae contribute to the attachment. If bacteria can better attach to the surfaces, is it more or less virulent? Speak up. More, meaning fimbriae, a virulence factor. Finally, pilus. Here you can see two pili, one pilus and another pilus, connecting two bacterial cells. Those are sex pili. They contribute to genetic exchange between bacterial cells. Does that make sense? You clear? So pili are unique in this case. They provide a direct way to transport genetic information between two bacterial cells. Now, few words about flagellar motion. There are two modes. It's called running and tumbling. So when microorganisms run, all flagella rotate in counterclockwise. Don't memorize counterclockwise. I don't really care. I want you to understand that running is directional movement. Tumbling, is staying in one place. So bacteria, they're not like Michael Phelps that just swim directly to the goal. They will swim, stop, turn, swim. So run, tumble, run, run, tumble, run, tumble, run, tumble. This movement can seem quite chaotic, but bacteria will swim, will run to increasing concentration of some kind of chemoattractant, usually nutrients. Does that make sense? They will swim towards the higher concentration of nutrients. And when they run, they swim along against the local gradient. They go from low concentration of nutrients to high. Does that make sense? And when they tumble, they stop and reorient themselves to find another gradient to swim towards. Does that make sense? So they're like chasing the highest concentration of nutrients. Are we clear? Now, I want to say a few words about the energy source for the flagellar rotation. There's one structure that is fairly common for both prokaryotes and eukaryotes. It's ATP synthase. If you taken some kind of basic cell bio, you had a conversation about cellular respiration. And in cellular respiration, protein that makes ATP is called ATP synthase. It works when hydrogen ions go across the membrane through that ATP synthase. The energy of hydrogen ions, kinetic energy, okay? It allows this enzyme to rotate and produce ATP when it rotates. So what bacteria do? They use some of the components of ATP synthase to rotate flagella. Bacteria do create a hydrogen ion gradient, just like they do in cellular respiration. Then, when hydrogen ions diffuse through the channel right here, okay, kinetic energy of hydrogen ions passing from high concentration to low. Look at this slope that I draw in the upper right corner. Think about this ball. It's a hydrogen ion. As it runs down the slope, it gains kinetic energy. Does that make sense to you? So same way, hydrogen ions, as they pass through this channel, they gain kinetic energy that is basically converted into kinetic energy of the flagella rotating. Does that make sense? Is that clear? Yeah. So that's I find it like fascinating because some parts of the flagellar motor are very similar to parts of ATP synthase. Okay? Good? Now, I told you that I'm going to give you a lot of fascinating examples. This is one of them. What can be a better example of bacteria? Only Staphylococcus aureus. Everybody heard about it. MRSA, Staph aureus, staph. You all know staph. So that's an infamous staph. So, to give an idea what I want you to know about pathogens that I introduce you to in general, I expect you to know the diseases that they cause, the transmission route, whether or not there is a vaccine, and if I specifically tell you, like, look, this is what you have to know, this is in the study guide, a virulence factor. For instance, we talked about Clostridium botulinum, right? And I told you that its toxin, Clostridium botulinum toxin, inhibits release of acetylcholine at the neuromuscular junction. You need to know that. I told you that titanospasmin, the toxin of Clostridium tetani, it blocks the relaxation of the muscles. You have to know that mechanism. And then I tell you, well, you know, in Clostridium perfringens, it kind of breaks down the cells, but it's not important. That's not important. Am I clear? All pathogens that we have vaccine against will be either stated on the slide, or if it's bacteria, there's going to be an asterisk to it. So if you go back to Clostridia page, you will see there is an asterisk next to Clostridium tetani. It means there's vaccine. Am I clear? Let's talk about staph. Let's get the boring stuff out of the way. Staphylococcus epidermidis. This, right. So this red growth, Staphylococcus epidermidis. And you have it right there, right? Staphylococcus epidermidis is non-pathogenic. It doesn't cause the disease. But, there's always but. In patients who are immunocompromised, it can. Patient has to be really screwed up. You have to understand that. Usually it's advanced AIDS patients with severe wounds, for instance. So it causes subcutaneous infections. In really, really immunocompromised patients, it may cause cardiac infection. But it's super rare. There are separate case reports. So it's not common at all. Staphylococcus saprophyticus can be found in the GI tract, can cause urinary tract infections. How can it get from the GI tract to urinary tract infections? To urinary tract, sorry. From GI to urinary. Huh? Yeah, bad hygiene. Yeah, bad hygiene. Also unprotected anal sex for males. That's the way, okay? So it's like strictly urinary tract infection. And here's our Staph aureus. So it causes few major diseases. We're going to start types of disease. We're going to start with most common. It's cutaneous disease. Cutaneous means that it's associated with skin, and I'm going to include stuff that's under. Probably the most common disease associated with Staph aureus on the skin is impetigo. People who have kids, work with kids, know. One kid has impetigo, all kids have impetigo. My kids played football in 2 months. Every kid on the team had impetigo. I don't know who started it, but it's transmitted by contact. You can imagine in the football, there's a lot of contact. So these lesions look pretty nasty, but they don't really hurt. They don't really itch. The treatment is, in kids, it's usually both topical and oral antibiotics. It's fairly easily treated. Okay. Now, in some cases, Staphylococcus aureus can cause cutaneous infections in newborns. Newborns have much weaker immunity yet compared to, toddlers or school kids. And in them, Staph aureus infection of the skin can lead to the condition known as scalded skin syndrome. Here you can see epidermis peeling off the hands of that child. Okay, it looks like a kid was showered with boiling water. Okay, this condition is very treatable. Usually it doesn't result in any further complications. The main risk associated with scalded skin syndrome is that secondary infections, in fact, can settle in, can get deeper into the skin or in the subcutaneous tissue. Am I clear? Getting deeper. Now, what's interesting, what contributes to the skin infections? I want to specifically make a point for exfoliative toxin. Basically, know it. Exfoliative toxin. You know, like exfoliative scrubs that remove old skin. Exfoliative toxin has a fascinating mechanism. It breaks down proteins that connect epidermis and dermis. Does that make sense? You can, I don't have this picture right here, but you can find a microscopic image, like a tissue slice. And it will show you, you have dermis, and then epidermal layer is separated from it. That's a scalded skin syndrome. Does that make sense? Okay. Many of Staphylococcus aureus strains are antibiotic resistant. That's MRSA, methicillin resistant Staph aureus. Now, another condition, well, I'll get to some other virulence factors later. Now, what if it gets deeper? If it infects hair follicle, it causes what's called furuncles. If several hair follicles adjacent to each other get infected, it can lead to that pretty nasty infection called carbuncle. Furuncles and carbuncles are often treated with topical antibiotics or surgically. When I was 11 years old, I had furuncle on my elbow. I went to the doctor. He told me, let me take a look. And while I showed him my elbow, I didn't see. He grabbed an instrument, stuck it right there, expanded it, all that bad stuff. Just basically he treated, he opened it up. No anesthesia. Just pure surprise. But I got treated. Now, in the worst case scenario, this microorganism can get even deeper into the muscles, leading to the condition that is called necrotizing fasciitis. Necrotizing means necrosis, fasciitis means fascia, connective tissue. Microorganism, Staph aureus, has enzymes that destroy connective tissue. And what is the function of connective tissue? To do what? Connective tissue. To connect. Connect, exactly, to connect other tissues. When other tissues are not connected, they practically fall off. This is what we call flesh-eating bacteria. It is notoriously hard to treat. If it's an infection of a limb, treatment are usually, well, massive systemic IV antibiotics, debridement, often amputation. Usually patients that are immunocompromised are most susceptible. I saw a presentation of clinical case, immunocompromised patient with diabetes who had necrotizing fasciitis on her trunk. She didn't survive because you can't amputate the trunk. Yeah. Huh? Well, you can, yes, but that's a form of capital punishment. Now, those are cutaneous, cutaneous infections. Another famous infection associated with Staphylococcus aureus is toxic shock syndrome. And it is caused by a virulence factor, a protein that is called toxic shock syndrome toxin. So what this protein does, it activates your immune system. Not just activates, it activates it over the top. So imagine this. If you ever got pulled over, one police car, right? Police officer comes over, checks your driver's license, whatever, okay? Now imagine that you get pulled over by the entire police forces of the city of Cleveland plus SWAT teams. That's not normal, right? And the rest of Cleveland has no, like, it's a disaster for the city. I don't know, whatever, like, people start firing at your car, they start throwing grenades at you, basically terrible stuff happens. This is superactivation of your immune system. When your immune response gets activated to that level, all your blood vessels become super permeable. So plasma starts to leak from the blood vessels in the tissues. What happens to the blood volume? It goes down. What happens to the blood pressure? It goes down. You experience hypovolemic shock. On top of that, inflammation leads to vasodilation. On top of hypovolemic, you have circulatory shock. So you have two shocks at the same time. And often you have disseminated intravascular coagulation. So in addition to these two shocks, your blood starts to clot everywhere. Pretty terrible story. First time, toxic shock syndrome, big outbreak, it was 60s or 70s, associated with the female hygiene products. There was a batch of tampons that was contaminated by Staph aureus. Got through the mucosal membrane into the circulation. I believe 72 women died because of that reaction. Okay, but the problem, it wasn't a synthetic material. It was still some natural product, like cotton or something. So it is a problem. So if Staph aureus gets in the blood, it can lead to this. And finally, if it's not enough for you, Staph aureus can lead to sinusitis. That's what I have right now. I tested myself last semester. MRSA is right there. I'm not kidding. It's MRSA. Huh? I'm colonized, yeah. Last semester, and I didn't have any symptoms. I had some mild rhinitis over the Christmas break. I'm fine now. So you can be respiratory colonized with MRSA, experiencing no symptoms. Percentage low. Yes, for Staph aureus, the normal percentage of respiratory colonization depends on the class. I had as low as 5%, as high as 20-25%. That's around it. MRSA is rare, but I did have a very interesting case when I had two students in two different classes. They were from two different cities around Cleveland, opposite sexes, never met each other, ever. They were sitting at the same place at the table. They both had MRSA in their throats. No symptoms, nothing, no disease, which is pretty cool. Now, sinusitis is common. It can lead to pharyngitis as well, so respiratory infections. In this case, MRSA can be transmitted respiratory, from person to person, by sneezing, coughing. If it gets in the blood and it gets into sterile locations, it can cause endocarditis, infection of the cardiac valves, or osteomyelitis, infection of the bone. Bone, yes. And we had first-hand experience with that. One of my kids broke the finger of another kid, also mine. And that kid with a broken finger had impetigo at that point. So Staph aureus sneaked into the fracture and established the infection there. So he had chronic osteomyelitis before it got detected, like half a year. It ended up being really painful, and he was on antibiotics for 6 months. So it's treatable, but in patients, for instance, in patients who are immunocompromised, osteomyelitis can be brutal and really hard to treat, because you can imagine delivering antibiotic to the bone is somewhat challenging. What always surprised me, we still have no vaccine against Staph aureus. So it's only treatment. Okay. And in relation to MRSA, at some point MRSA was, oh my God, we've got MRSA, we're all going to die. It's a emerging pathogen, super terrible. I listened to the interview with the head of emergency department, some hospital in New Jersey. Said MRSA, it's new normal. Well, it's there, it's out there. We just have to presume that multiple infections, may have MRSA component to it. Any questions? So we've got nice little wrap up with bacteria. We're not going to move any further, as you can imagine. I will see